to Miss Johnson's Biology News for Odd. I am Claire Anderson, your resident host for this afternoon, and I have a wildly fascinating discovery that has just surfaced in the last week. On April 19th of 2018, researchers released evidence of genetic mutations in South Asian divers who have acquired enlarged spleens due to generations of diving for their food. For thousands of years, the Bajau people, a people of marine nomads, have survived by diving for fish and seafloor creatures for sustenance. The divers spend up to 60% of a workday underwater, so for a long period of time, the scientists considered their astounding ability to hold their breath to be physically acquired. For divers, the spleen is an incredibly important organ that stores oxygen-rich red blood cells and releases them into the bloodstream. The first study conducted on spleen mass and maximum diving time was through a study of seals, and through this study, a direct correlation was found. This connection, however, had not been researched on the genetic level and this is where our researchers found their opportunity. Researchers discovered that the enlarged spleen size was likely a genetic mutation after comparing the spleens of diving and non-diving Bajau people, along with comparing the Bajau to their neighbors, the Saliwan people. When analyzing the spleen inside the Bajau, the enlarged spleen was found not only in the divers, but in the non-diving members of the community, showing scientists that the spleen size is likely not acquired by training underwater, but by genes. The genetic mutation theory was strengthened by the fact that the Saliwan people had normal-sized spleens, unaffected by living in a South Asian marine environment. A gene mutation was indeed found to be acquired by the Bajau people, gene PDE10A, resulting in the Bajau people having a larger spleen. This genetic discovery has offered great promise in hypoxia studies by studying the Bajau people's genetically acquired hypoxia tolerance. Hypoxia is a lack of oxygen to the bloodstream, a condition that occurs at high altitudes and deep waters for a long period of time. The Bajau's tolerance will help scientists research hypoxia in relationship to cell volume, thyroid, and spleen size. This discovery is an amazing example of the power of evolution and a recent example of human evolution.
Hello viewers, and welcome to Miss Johnson's 4 Odd Biology in the News. My name is Alyssa Barber, and I will be your host. Today we will be talking about the limb usage of marine tetrapods, specifically marine turtles. Marine tetrapods are a group of animals that have adapted to live on land, then sea, back and forth over and over again throughout the last 350 million years. Their migration from land to sea and sea to land has required an extensive adaptation in short periods of time, making them very unique animals. A study done by a research team made up of 5 scientists, Jessica A. Fuji, Don McLeish, Andrew J. Brooks, John Gaskell, and Kyle S. Van Houten, looks deeper into the extent of marine tetrapods having evolved to use their limbs. In this study, the team of researchers collected data on many marine tetrapods, looking at evolutionary, morphological, ecological, and behavioral factors to better understand the pattern of limb use across marine tetrapods. However, the most interesting part of the study was looking at limb usage in sea turtles. By looking at underwater surveys, web image and video searches, and published literature, the scientists found that sea turtles use their flippers for more than just swimming. For the purposes of the study, the actions the sea turtles performed were categorized as holding, digging, striking, tossing, leveraging, swiping, corralling, and pounding. In the research, the turtles are pictured karate chopping jellyfish, rolling scallops along the seafloor, and grasping coral in order to remove a sea anemone, among other things. The study primarily looked at green turtles, loggerhead turtles, and hawksbill turtles. Hawksbill and green turtles were observed using corralling, leveraging, holding, and swiping movements to capture, process, or transport a variety of prey, while loggerhead sea turtles were seen using swiping to process their own prey. The feeding techniques described were not all that common, and the researchers believe that this could suggest that these techniques are not used to expand the foraging habits of sea turtles, or possibly only used in specific niches not used day-to-day by sea turtles. Nonetheless, any regular limb use beyond that of swimming for sea turtles could promote these other foraging tactics used by sea turtles. For example, female sea turtles return to land to lay their eggs and use both fore and hind limbs to dig body pits and nesting sites. The biggest factor in the development of limb use in these marine animals may be the overall limb mobility of the animal. The wide array of foraging tactics in sea turtles suggests that their flippers are very mobile and are not constantly needed for propulsion, allowing them to use their limbs for other purposes. How the sea turtles began to use their flippers by foraging instead of just swimming is unknown. The researchers did not expect to see such a large array of different actions done by sea turtles using their flippers for several reasons. Sea turtles are never taught by their mothers to forage, as they are on their own as soon as their mother lays the eggs on a beach. The flippers are also highly specialized for swimming and are not as well suited for other uses. The researchers also did not expect to see such complicated actions due to the small size of the sea turtle's brain. Overall, the study suggests that these limb use behaviors across marine tetrapods are limited by mobility, and that the frequent use of forelimbs for other behaviors may promote the development of these feeding strategies. These observations provide additional insight into the diversity and possible evolution of limb use behaviors throughout marine tetrapods. I'm Melissa Barber, finishing off today's Bio in the News. Thank you for tuning in.
Thank you for joining us today for Ms. Johnson's Biology in the News. I'm your host, Jacob Badcock, and today we will be talking about new discoveries in Malu Basin, Sabah, Borneo. On April 30th, Taxon Expeditions, an organization that involves citizens in taxonomic discoveries, released results for the first taxon expedition. These results revealed the discoveries of 3 new specimens of the genus Gravelinus. The first of these 3 was Gravelinus quest. This insect resembles Gravelinus anus. Quest can be identified by its 550 to 560 micrometers long AD and ventrally bent tip of the median lobe. It was discovered at the creek east of Nepenthes Camp. The second species discovered was Gravelinus andrecuipersi. Andrecuipersi resembles Gravelinus theonmacjac and Gravelinus sumatranjac. Andrecuipersi can be identified by its pale elytral patches, smooth elytral surface, and a smaller aegis with distinctly varying base, median, and perimeters. It was discovered upstream Gillick Falls. The last of the 3 new specimens was Gravelinus Leonardo DiCaprio. Yes, you heard the name right. This beetle was named by the citizens who discovered it after the actor Leonardo DiCaprio to honor his commitment as an environmentalist. It can be identified by its large splendor elytra and the fully glabrous and not elevated median pronotum. The citizens found this beetle upstream Gillick Falls. Thank you for joining me today as we discuss the new 3 species discovered in Borneo.
Hello, I'm Wes Brownlee and welcome to today's segment of Biology in the News. Today we'll be discussing how zebrafish are able to completely rejuvenate their damaged tissues. A recent study conducted in Japan by Professor Atsushi Kawakami and graduate student Iri Shibata shows that zebrafish are able to completely regenerate their lost tissue through stem cells located in the basal layer of their skin. During the experiment, the research team used the fins of zebrafish, missing fin, that had tissues removed, and they were studied to show how the stem cells in the basal layer of the skin could regenerate the damaged tissue. They labeled the rejuvenative tissue with fluorescence using a genetic cell labeling technique called Crelox-P, and tracked how the cells were progressing over the course of the next few weeks. In the study, the first group of epithelial cells would appear on the site of the wound but would disappear a few days after due to the apoptosis. The second group of cells arrived and they would begin to rejuvenate the damaged tissue. It was discovered these epithelial cells came from the large area of the skin containing the stem cells. Researchers involved believe that this rejuvenation could be possible in humans if we're able to control the autonomous proliferation of stem cells in the basal layer of human skin. If the mechanisms that zebrafish use are the same as those that humans use to heal, it is expected to lead to the causes of many skin diseases.
Hi, this is Kishu Chaudhary and welcome to this segment in Biology in the News. Summer is just around the corner and CDC experts are advising all, beware the bugs. Illnesses from tick, mosquito, and flea bites have tripled between 2004 and 2016. Since 2004, 9 new vector-borne pathogens have been identified. These vectors are blood-feeding insects that spread their pathogens like bacteria, viruses, and parasites by biting into human skin, causing severe illnesses. According to the World Health Organization, vector-borne disease accounts for 17% of all infectious diseases, causing 700,000 deaths a year globally. A new report based off the data from the National Notifiable Disease Surveillance System identified 16 different diseases between 2004 and 2016. Of those 650,000 cases, tick-borne illnesses accounted for more than 75% of reports, significantly impacting the eastern part of the United States and areas along the Pacific coast. Of the cumulative reported tick-borne diseases, 82% accounted for Lyme disease. Mosquito-borne diseases have been more dispersed and more stressed by epidemics. When the Zika virus outbreak occurred in 2015, a big jump in mosquito-borne diseases occurred as nearly 88% of the cases reported in 2016 were related to Zika virus. Another common mosquito-borne disease transmitted in the United States was the West Nile virus, significantly during the 2012 outbreak that impacted Texas. Flea-borne diseases were rare, not exceeding 17 cases a year, as plague was the most common disease that was transmitted, primarily in the rural southwestern part of the United States. So why are vector-borne disease cases rising? Ticks thrive in dense forested areas as suburban communities continue to expand their habitats. With rising temperatures, this allows ticks to migrate further north and extend the tick season. As for mosquitoes, us people and goods are moving around the planet at increasing rates, as many mosquito-borne diseases can be transmitted almost anywhere in the world. Travel and trade are two factors that increase our risk to the admission of exotic mosquito-borne diseases. Note that this study had some restraints, as these vector-borne cases included data only reported to public health officials. Nonetheless, the study shows how vulnerable we humans are if a sufficient amount of vectors are present and the population is not immune to the disease. Thank you for joining this segment in Biology News.
Good afternoon. My name is Rachel Dishman, and welcome to another segment of Miss Johnson's Biology News Network. Scientists from Sidney Garvin's Institute of Medical Research have discovered how antibodies that were thought to be bad for the immune system actually have a positive purpose, a purpose that saves people from invading microbes. These antibodies were originally thought to react against the body's tissue and damage the autoimmune system, so they were made inactive to prevent any more damage to the body. Scientists, though, were still puzzled as to why the body did not just destroy these antibodies while they were silenced. Co-leader of the experiment, Professor Chris Goodnow, was baffled by the results and said that people's perception to how the immune system protects us will be changed forever. These once believed bad antibodies go through a process that changes them from bad to good. Once this process is over, they are able to fight the invading microbes. The silent cell army contains millions of immune cells known as B cells. These cells produce antibodies to fight diseases. Selected cells in the army can be reawakened to fight invaders, but only once their bad antibodies are made good. When testing to see how these antibodies redeemed themselves and became beneficial for the body, scientists need to see how the DNA of the antibodies was altered. They found out with their mice model system that only 3 DNA changes were needed to transform the antibodies. The first change is to stop the antibody from damaging the host. The next 2 changes are to increase their ability to damage the invader instead of their host. The original good antibodies are no match for the invading microbes that try to destroy the immune system because these bad diseases disguise themselves to look like they are beneficial for the body. By having the background of being a bad antibody, the bad antibodies know the signs to look out for when they see something new come into the body, pretending to be beneficial, and they can immediately attack them and prevent the host body from suffering. Diseases like Campylobacter and HIV are examples of diseases that look identical to the body's molecules and usually get missed, causing people to suffer. With the discovery of these new antibodies, though, new vaccines are being made and tested in mice to find a way to prevent these horrible diseases from destroying immune systems and the human body. Scientists are now processing this new information and using the B cells to create vaccines against HIV to hopefully find a cure. Thank you for listening to this segment of Miss Johnson's Biology News Network. Have a great day!
Hello all, this is Marshall from Miss Johnson's Biology in the News. A new study has recently come out from the UK surrounding the loss of respiratory ability in European mistletoe, specifically Viscum album, due to an increased reliance on parasitism. The study talks about how the absence of complex 1 is associated with the diminished respiratory ability of the mistletoe. Complex 1 is the first collection of proteins in the electron transport system, otherwise known as NADH dehydrogenase. This is a major finding because without the NADH dehydrogenase, the activity of the rest of the electron transport system is reduced. Specifically, the study found that the activity in complex 4 and ATP synthase was reduced 5-fold, meaning that the mistletoe has been converting to parasitism instead of going through the normal function of the mitochondria. One of the main reasons this could be an interesting new study is that it is the first time a multicelled eukaryotic organism has lost the ability to produce complex 1. The mistletoe is losing its ability to produce complex 1 because those that had already lost their ability to produce complex 1 were able to reproduce faster, leading it to taking over the population. While this trend is new to multicell eukaryotic organisms, it has previously been seen in single-celled eukaryotic organisms and prokaryotic organisms. It's part of a process generally referred to as reductive evolution. Reductive evolution is when the organism evolves or changes by losing genes instead of changing or gaining new ones. To make up for the loss of NADH dehydrogenase and the reduction in the ETS, the mistletoe instead ramps up the activity in glycolysis and other processes that operate in a similar vein. Even though glycolysis and other cytosilic reactions are increased, the Krebs cycles does not see the same increase in activity, most likely having something to do with how Krebs cycle is mostly used to produce NADH, something which is no longer really used due to the lack of the electron transport system. To make sure that the study was accurate, the researchers took samples from 2 different populations, one in Germany and one in the UK, and both locations found the same results. In summary, the study looks at the vast change in the function of the mitochondria in European mistletoe due to the difference in the electron transport system and it lacking. Now for the next news story.
Hello, and welcome to the next segment of Bio in the News. A new study recently published looks at the effects of antibiotics on mammals born prematurely. The study focused on how early microbial colonization affects DNA methylation of genes related to intestinal immunity and metabolism in preterm pigs. It looks specifically for regions with differing methyl groups and different levels of gene expression. Pigs were used in the study because of the high similarity of intestinal microbiomes and DNA methylation processes in pigs and humans. The study consisted of 2 groups, one of which was given antibiotics in the first 5 days of life to postpone bacterial colonization, and a second group which was allowed to come into contact with beneficial bacteria. It is common in the medical field to let premature humans come into contact with beneficial bacteria while still being protected from pathogens, in accordance with a philosophy called the hygiene hypothesis. This hypothesis states that exposure to bacteria and other microorganisms is required for the healthy development of the immune system and is supported by many other studies. Samples were taken from the intestines of both the antibiotic group and the control group so that DNA methylation, bacterial biomes, and the like could be observed and compared. Compared to the control group, pigs that were given antibiotics had 47 different regions on their genome that were hypomethylated. This change in methylation can affect gene expression, and these specific hypomethylated regions can have effects including decreased innate immune response, reduced glycolysis rate, and increased gluconeogenesis-related gene expression. One possible reason for these effects is the use of bacterial products like fermentation metabolites that are used indirectly by the body to help maintain normal function and development. Another reason for these effects could be that since the body does not come into contact with invading bacteria, it does not need to launch an immune response, and so there is less development in that area. One beneficial effect of the antibiotics was a lowered level of hypoxic stress. This is because the reduced expression of a gene involved in a metabolic pathway that works to compensate for decreased oxygen levels. Another major benefit for the antibiotic group was the successful prevention of necrotizing enterocolitis, which is a disease that affects about 10% of all hospitalized preterm infants. Antibiotics treatments like the study are being tested as a possible way to prevent this disease. However, while the treatment was successful in preventing this disease, it was also correlated with the lower innate immune response and increased metabolisms. This study greatly furthered our understanding in the area of mammal development and DNA methylation methods, but further research is needed to understand these complex processes. Thank you for joining us in this segment of Bio in the News.
Good afternoon, my name is Madison Lester, and welcome to today's segment of Johnson's Biology in the News. In today's study, we are going to look at germ layer commitment and the axis formation in sea anemone embryonic cell aggregates. The sea anemone are important parts of the marine ecosystem, forming many mutualistic symbiotic relationships with the organisms around them in their environment. The
sea anemone Nematovecinus have embryonic cells where the aggregates can restore germ layers and fix the axis patterning of the cells. A group of marine researchers conducted an experiment with segregated endodermal cells and ectodermal cells to determine which ones, if any, would be able to change the form and aid the germ layer commitment and axis formation. The endodermal cells and the ectodermal cells were dissociated and then left alone to track the normal cells of the aggregate development of the sea anemone shown in figures A through H and I through T. After this data was taken and analyzed, the team cut the wild-type gastrulae in half, splitting up the oral, which is the end that is closest to the mouth, and the aboral end, the closest away from the mouth. When separated, the oral halves turned into the normal polyps, figures A through E, and the aboral halves turned into ciliated balls lacking the endodermal cells, figures F through E. Next, the aboral halves of the wild gastrula and the aboral halves of the gastrula expressing the life act and orange 2 glowing cells were put together in figures K through N. The cells are seen dispersed to the polyp, existing both oral and aboral ends. Next, the researchers combined the ectoderm in the wild type and the endoderm expressing life act and orange 2 in figures O through R. These fluorescent cells all migrated to the inside, seeing as they were all endodermal cells to begin with. Another test including aboral half of the wild type and the endoderm expressing the life act MRH2, which formed a polyp with fluorescence in the endoderm but without the normal organizer cells seen in figures S through B. The final test in this group was with the endodermal layer figure W through Z, and the endoderm without the ectoderm layer became mesenchymal and left the aggregate forming the splatter of cells seen in figure Z. The scientists then used a different fluorescence in cherry with the wild type to further look at tracing the endoderm and ectoderm through the formation of the polyp. In figures A' through E', the blastopore lip ectoderm of the wild type and the aboral half of the M cherry ectoderm were combined as seen in the pictures to my right. The M cherry was not expressed until C' when it's very visible. Eventually, in figure E', some glowing endoderm cells can be seen in the form of polyp. However, when the oral half of the M cherry gastrula was allowed to grow on its own, the glowing endoderm cells were seen throughout the fully formed polyp. All of the previous tests were conducted to see the changes in aggregate formation when all of the original cells were not present. To test the germ layer and commitment and axis formation of the sea anemone, the scientists used their knowledge of WNT1 and WNT3 and the BMP signaling to figure out whether or not the anemone could do the known things. Two aboral halves of the gastrulae with one containing WNT1 and WNT3 plasmids were combined. The aggregate, not originally having endodermal cells, was able to form a primary polyp, concluding that the cells WNT1 and WNT3 plasmids were able to gain the ability to repair germ layer and keep the axis formation, as well as inducing self-organization of embryonic aggregates in figures A through C. Looking into another factor of axis formation, the BMP signaling determines whether or not an aggregate will have direct axis or no axis at all. A BMP signaling component, BMP24 morpholine, blocks the organism's ability to have direct axis. Aggregates with BMP24MO knocked down successfully forms a polyp, but the polyp lacks any distinct axes as expected in figures D through F. Nematostella is used to uses an alternative development trajectory to respond to the lack of cavity in an aggregate, or the prevention of invagination due to using delamination of the ectoderm layer to form germ layers. The placidity in the sea anemone is not a trait of just early branching metazoan embryonic cells. Similar placidity has been found in sea stars, sea urchins, and echinoderms. Alternative development trajectories are proposed to be easily attainable to organisms without highly mosaic development, also suggesting that the plasticity of the embryonic cells is mostly found in the beginning phases of the evolution of animals. The ability to adjust the embryonic cells development without destructive consequences could have aided in the diversity of formations of animal-bodied plants, further in aiding in evolution of animals. Thank you for joining me for today's segment of Johnson's Biology. Now moving on to the next topic.
Hello and welcome to today's episode of Miss Johnson's Biology in the News. I am your host, Shannon McClandrick, and I would like to touch on a new story that has surfaced in the past couple of weeks. Researchers have potentially found a new treatment for cancer by targeting certain proteins called proteasomes. Proteasomes are complexes made of proteins that work to degrade other proteins in a process called the ubiquitin proteasome pathway. In this pathway, proteasomes degrade several proteins that are in charge of processes such as translation and apoptosis, which causes the mutation of cells and allows for diseases to appear, such as cancer. In essence, proteasomes play a huge role in the development of cancerous cells. With that said, how do we stop these proteasomes from breaking down too many proteins? In a study conducted by Jose Rodriguez and Erman Steller, they tested the effect of inhibitors on the assembly of 26S proteasomes in a colorectal tumor. They used mice and intestinal tumors that were induced into them to give them cancer. They then used the inhibitor PSMD5 to see how it would affect the translation rate of the proteins in the mice cells. The study's results showed that the protein translation was increased in the intestinal tumors with the use of the PSMD5 inhibitor. This caused the inhibitor to be silenced in the beginning, but once the inhibitor PSDM5 was re-expressed, it caused a decrease in the amount of 26S proteasomes and also decreased the accumulation of ubiquinated proteins. Overall, studies such as this have sparked ideas of creating a new way to treat cancer by eliminating the amount of proteasome assembly in the body. The evidence supporting the use of proteasome inhibitors is enough to suggest that more studies need to be done in order to create a fully functional method to target cancerous cells in humans. Although we may be far from trying a new method, we definitely have a strong start to saving millions of lives in the fight against cancer. Once again, I am your host, Shannon McClandrick, and thank you for tuning in today.
Good afternoon, and welcome to another segment of Johnson's 4 Odd Biology in the News. Today we'll be addressing the latest discovery of a new DNA structure within living human cells. The structure, known as the intercalated motif, or I motif, while it had been previously discovered in the 1990s, was recently found in living cells. The I motif is a 4-stranded knot of DNA. Where the helical form's nitrogenous bases pair oppositely, the I motif's C's pair with each other, not to their corresponding G's. Initially, the I motifs were only witnessed in vitro, or under artificial conditions in the laboratory. In order to detect I motifs inside cells, Dr. Daniel Christ and Dr. Marcel Dinger developed a tool that could specifically recognize and attach to I motifs. The tool, which is a fragment of an antibody molecule, is unable to detect DNA in helical form, nor can it detect G quadruplex structures, making the process of detecting I motifs significantly easier. By using fluorescence techniques to distinguish where the I motifs were located, numerous green spots were identified within the cell's nucleus, therefore indicating the presence of I motifs. The scientists were then able to show when the I motifs tend to form in the cell's life cycle, the late G1 phase, where the DNA is being actively read. They also showed that I motifs can appear in areas of the DNA that control whether genes are switched on or off, or promoter regions. They also appear in telomeres, the end sections of chromosomes. It isn't yet clear what I motifs do, but study author Dr. Mahi Zerati believes that the coming and going of I motifs is a clue. It seems likely that they are there to help switch genes on or off and to affect whether a gene is actively read or not, says Dr. Zerati. Dr. Christ adds, we also think the transient nature of the I motifs explains why they have been so very difficult to track down in cells until now. This extraordinary find could pave a new path to understanding this form of DNA and what it can do, as well as how it could impact health and diseases. This has been Kellen Miller with the latest biology in the news. Have a good day.
Hello, and welcome to another segment of Miss Johnson's 4 Odd Biology in the News. I am Mitchell Natali, here to give you the latest in biological news. Today we have some explosive news for you from Southeast Asia. Scientists performed field research in the Turenberg district in Brunei, collecting specimens of ants they believed to be a part of a new species. Their research found to be successful as they identified a new species called Colobus explodens. From the collected specimens, the scientists extracted DNA from minor workers and major workers of the ant colonies using their limbs. The molecular data showed that this species was in fact entirely different than the other 5 species that were previously known. The species name for them is known for their unique behavior called autothesis, found in other species of ants in other parts of the world, but also in termites. This is when they sacrifice themselves to protect the rest of their species. To defend the colony, minor workers will raise their gaster in a defensive position. When they feel the threat is sufficient, they will contract their gaster until the integument ruptures. This will release a sticky, slightly viscous, bright yellow irritant that is effective in warding off other arthropods but ineffective in warding off other large animals. Though this may kill the worker, this is for the benefit of the colony and shows the dedication of the ants to their queen. Talk about motherly love taken to a new level. This fantastic species of ant is just another example of how diverse our planet is. Thank you for tuning in. This has been Mitchell Natali for this segment of Biology in the News.
Hello everyone, and welcome to today's Bio in the News segment. A recent study has found that after multiple generations of fish were exposed to a warmer ocean by 3 degrees Celsius, they are able to adapt to the stress through phenotypic plasticity. This is the ability to express multiple phenotypes for one genotype, designed to protect against environmental changes in a way that is quicker than genetic adaptation. Specifically, transgenerational acclimation, a form of phenotypic plasticity, occurs when the environment that parent organisms live in affects the gene expression of their offspring. Although the exact mechanism for phenotypic plasticity is unknown, scientists suspect DNA methylation to play a role in this transfer of epigenome information. Scientists sequence and analyze the liver genome, methylomes, and transcriptomes of the coral reef fish Echinochromis polyacanthus to different water temperatures. Fish exposed to the temperature increase indicated an altered epigenome through DNA methylation of specific loci. Those exposed through multiple generations express genes differently through more genes by greater magnitude than those exposed for just one generation. Functions related to energy homeostasis, such as insulin response, nutrient metabolism, and mitochondrial activities, were prominent among the differentially methylated genes. This indicates that transgenerational fish have a more energy-efficient response than that of fish directly exposed. Scientists identified 193 differentially methylated genes that correlate to aerobic skill, an organism's ability to provide oxygen above metabolism requirements. Key ones involved include metabolic homeostasis, insulin sensitivity, and improved oxygen delivery, which show a strong association with physical acclimation to heat stress across generations. With exposure to higher temperatures due to climate change, genome changes in DNA methylation show the strong role transgenerational acclimation could play in reef fish facing warming oceans.
Hello, my name is Gabby Oliveri, and I will be your host for today's segment of Johnson's Biology in the News Network. Today, we will be discussing how microbes in underground aquifers chow down on carbon. Everything on Earth, from ants to humans to sharks, uses carbon to survive. However, not all carbon is the same. In the oceans, some carbon-containing compounds, such as sugars and proteins, are quickly utilized by microorganisms, while others, like chitin and fish scales, are much harder to consume. On the slide behind me, microbes in an aquifer feast on carbon flowing through a permeable rock. Many scientists believe that a specific type of carbon called refractory carbon was not eaten by any organisms and simply made up the sediment on the ocean floor. Ecologists Sunita Walter of the University of Delaware and Peter Gerges of Harvard University have now discovered that underground aquifers near the underwater mid-Atlantic ridge pull in oxygenated seawater and allow microbes to consume more refractory carbon than previously thought. This shows that microbes can be fed by seawater circulating through deep ocean crust, and microbes change the composition of the seawater that circulates back to the ocean. In the slide behind me, 2 pictures display the ocean carbon cycle, which shows one way how carbon enters into the ocean by diffusing into the surface waters and dissolving. While Gerges and his colleagues were able to show that microbes in the aquifers could eat carbon, it remained pretty unclear how active these microbes were. Scientists have been able to show that water comes in with a certain oxygen concentration, and as it drops, the carbon goes down in direct proportion. In the future, scientists hope to better quantify how much carbon is being consumed as seawater moves through aquifers. This was Gabby Oliveri, and I hope you enjoyed this segment of Johnson's Biology in the News. Thank you.
Hello, and welcome to Biology in the News. My name is Angel Pachimola, and today I am bringing you the latest in cancer research. In March of 2018, the National Institutes of Health released interim results from the 3rd phase of an NIH-funded clinical trial. The trial studied the treatment of patients with desmoid tumors, also known as DTDF, with the drug sorafenib tosylate. A desmoid tumor is an abnormal growth that arises from connective tissue, which is a tissue that provides strength and flexibility to structures such as bones, ligaments, and muscles. Typically, a single tumor develops, although some people have multiple tumors. The tumors can occur anywhere in the body. Desmoid tumors have similarities to fibrosarcoma, which are malignant or cancerous. However, desmoid tumors are considered benign or harmless with the exception of some symptoms because they do not metastasize or spread to other parts of the body. Because of the nature of these tumors, they are rarely considered fatal, but in some cases where the tumors grow rapidly, the masses can grow into nearby tissue and become fatal. Furthermore, the drug sorafenib-tosylate, or Nexavar, its brand name, is a form of chemotherapy taken by pill that can treat kidney, liver, and thyroid cancer. The drug was provided and developed by Bayer Healthcare AG. Sorafenib is a tyrosine kinase inhibitor anti-neoplastic agent. That means it has the ability to block the action of enzymes called tyrosine kinases. Tyrosine kinases are a part of many cell functions, including cell signaling, growth, and division. These enzymes may be too active or found at high levels in some types of cancer cells, and blocking them may keep cancer cells from growing. Sorafenib specifically interferes with the growth of tumor cells and new blood vessels that tumors need to grow. So, the third phase of the clinical trial was double-blind and had 87 participating patients during March through December of 2016. To be eligible for the trial, patients had to have DTDF that could not be surgically removed, tumors that had grown, or certain symptoms that they had to meet, and they had to meet other specific criteria. Patients were randomly assigned to receive either 400 milligrams of sorafenib a day or a matched placebo. Patients assigned to receive sorafenib were more likely to experience symptoms and side effects that were drug-related than patients assigned to receive the placebo. Nevertheless, side effects like fatigue, rashes, gastrointestinal infection, hypertension, and vomiting were not generally severe. Treatment to all patients continued until their tumors grew or less advantageous events occurred due to treatment. Patients whose tumors progressed were informed of their assigned treatment, and if they had been receiving a placebo, they were allowed to receive sorafenib if warranted and after consulting their physicians concerning further treatment options. The main goal of the clinical trial was to reach progression-free survival. Progression-free survival is the length of time patients lived before their disease worsened. The target improvement for patients given sorafenib was 15 months, while for placebo patients it was 6. After analysis of interim results of the first 75 participating patients in the trial, the improvement in progression-free survival exceeded the targets. This clinical trial shows the major progress and success researchers, scientists, and health specialists have made in cancer treatment today, as well as the potential of alternative treatments and future research that may eventually lead to a cure. Full detail and results from this trial will be presented at an upcoming scientific meeting and in a peer-reviewed publication. Keep up with this trial and all the most current updates in cancer research by subscribing to the NIH email list and following them on Twitter. I'm Angel Pachmola, and thank you for joining me with this segment on Biology in the News. Stay tuned for more on the latest in biology.
Hello, my name is Andrea Parrish, and welcome to another segment of Biology in the News. This episode, we will be discussing an experiment by Japan's University of Health and Welfare. The experiment was done to see if the opinions of people eating together affects one's taste threshold. For this experiment, they used 12 young women whose average age was 18 as their sample. These participants ate pieces of white bread in 3 conditions. The first one was that the participants ate bread with 3 quiet strangers as the control. The second one was in a positive condition, where the participants ate bread with 3 strangers who talked favorably about the taste of the bread. The third one was where the participants ate with 3 strangers who talked negatively about the taste of the bread. Each of the 12 participants had no food 2 hours prior to testing and tasted the bread in each condition 3 days apart. Participants also washed out their mouths with ion-charged water both before and after tasting. Immediately after tasting the bread, they completed an interpersonal reactive index, or IRI, where they completed a questionnaire that measured their opinions about the bread on 5 main categories: deliciousness, intensity of taste, texture, taste palatability, and appetite. These measures were then compared to the 3 conditions, and a ANOVA test was conducted to find the p-value. The results of the 5 measures differ significantly between the quiet condition, negative condition, and positive condition. As the graph behind me shows, The scores for deliciousness, intensity of taste, texture, taste palatability, and appetite differ significantly lower for the negative condition than compared to the quiet and positive conditions. Therefore, the study concluded that the taste can be influenced when eating and talking with others. The experimenters plan to further study the ways that taste and intake of food is influenced by one's environment by looking at the relationship between cooking and conversation. That concludes this edition of Biology in the News. Join us next time for new, more interesting stories in biology.
Hello and welcome to Ms. Johnson's 4th Odd Bio in the News. I am your host, Caroline Patey, and today we will be examining a new breakthrough in neuroscience. Formerly, CTE, or chronic traumatic encephalopathy, was associated with repetitive concussive experiences. However, recent research suggests that the mechanisms that cause concussions to take place may be independent of CTE risk. To better clarify, I'll explain a little bit about what CTE is. CTE is a neurodegenerative disease caused by a buildup in tau proteins in the cortical sulci. To better understand the cause of this, Boston University conducted an experiment that involved the use of an impact machine on mice to induce closed head impacts that simulates a mild head injury in which concussive-like symptoms were observed shortly afterwards. This was to be expected, but the researchers also conducted the experiment through the use of a blast on the mice, which did not cause them to experience concussive-like symptoms. However, both of these levels of the independent variable caused traumatic brain injury and displayed early symptoms of early pathology and sequelae that are associated with CTE. This discovery concludes that the risk of this neurodegenerative disorder may be much larger than originally thought. Thank you for watching and have a great day.
Welcome to another segment of Biology in the News. I'm your host, Catherine Queering. Today we will be discussing the initiation or early stages of post-materialization in the blue crab. All blue crabs have tough exoskeleton and must molt their shell periodically throughout their lifetime. Once the molt starts to begin, the classification is affected by the biochemical changes from their original matrix. After the molt, sugar moieties are enzymatically altered on cuticular glycoproteins secreted by the epithelium. The first mineral deposited is the amorphous calcium carbonate, ACC. Before the mineralization of ACC, silicon is seen in the areas of the exoskeleton about to mineralize. As the calcification continues, the silicon is no longer detected. A team of researchers hypothesized that silicon is involved in the stabilization of ACC by destabilizing the crystal lactase of calcite before it undergoes the transition to or is overgrown by crystalline calcite. In the picture, it shows how calcium and silicon is within the carapace and arthrodial membrane. The labels on the left specify the molt stage and the tissue type. The results revealed is that the silicon levels were equal to the calcium and carapace before mineralization. Once the mineralization began, silicon levels decreased and the calcium levels increased. In each graph, it represents the ratio of calcium to silicon in which stage of the molting is happening. In graph A, it shows the atomic percent of calcium and silicon present in mineralization. The other graphs show the increase and decrease of silicon and calcium throughout the stage of mineralization. In conclusion, silicon and calcium are very important in mineralization because these chemicals form the foundation of the new mold in the blue crab shell. Even though silicon is only present at a detectable level in the mineralizing tissue and is absent in all the stages observed in the non-classifying arthrodial membrane cuticle, more research is being conducted today in order to find why the silicon levels increase and decrease at different times during the molting of their exoskeleton. I'm Catherine Queering, and thank you for joining me on the Biology in the News Network.
Good afternoon. This is Noah Smith reporting live from Ms. Johnson's Biology in the News Network. Are you intrigued today by fast-growing solar panels and power technology? Things such as solar-powered cars, planes, and houses that have more commonly popped up in our modern world? Well, recently another new finding for solar has popped up, except it's not technology, it is set in an animal. According to the new study at Rutgers University, scientists have discovered a nearshore northeastern sea slug that has gained their own form of solar-powered technology. They have started to suck raw materials from algae to provide a lifetime supply of solar energy. These animals become plant-like after pilfering tiny solar panels and storing them inside the guts from the algae. Debashtis Bhattacharya, the senior author and distinguished professor in the Department of Biochemistry and Microbiology Engineering at Rutgers New Brunswick University, states, these slugs, Elysia chlorotica, steal millions of green-colored plastids from the brown algae, Valturia blatoria, and the waterproof pools near their feeding environments. These mollusks can grow up to more than 2 inches long and have found in the intertidal zones between Nova Scotia, Canada, Massachusetts, and Florida. Photosynthesis is when algae and plants use sunlight to create chemical energy, sugars from carbon dioxide and water. The brown algal plastids are photosynthetic and organelles like the organs in humans and people with chlorophyll. Bhattacharya states the particular algae is ideal for a food source because it does not have walls between adjoining cells and its body, essentially a long tube with located nuclei and plastids. When the sea slug makes a hole in the outer cell wall, it can suck out the cell's contents and gather the algal plastids all at once. Based on the studies of other sea slugs, some scientists have argued that they steal and store the plastids as foods to be digested during hard times, like camels store fat in their humps. However, Bhattacharya states this study has shown that it's not the case for these solar-powered sea slugs. The reporting that it has a remarkability to steal these algal plastids, stop feeding, and survive off the photosynthesis from the algae for the next 6 to 8 months. The team of Rutgers and other scientists used RNA sequencing and gene expression to test their solar energy hypothesis. The data shown that the slugs respond actively to the stolen plastids by protecting them from digestion and turning on the animal's genes to utilize the algal photosynthetic product. Their findings mirror those found in coral as maintaining dinoflagellates, also an algae, as intact cells and not stolen ones, in a more symbiotic relationship instead. Whereas Elysia chlorotica stores plastids, the algal nuclei are also sucked and don't survive. The scientists still don't know how these sea slugs maintain the plastids and the photosynthesis for the months without the nuclei that are normally needed to control the functions, Bhattacharya states. The slugs eat the algae to absorb its chloroplast, the organelles that actually do the photosynthesizing. But in order to turn the sunlight into chemical energy, the chloroplasts need a set of specialized proteins to help. So the slugs lift the genes from the algae that allow it to make the protein itself. Scientists don't know yet how this happened, but these sea slugs then pass their algal genes onto their babies, which only need to eat about 2 weeks out of the whole year, and the rest of the time they're soaking in UV rays. The broader implication is in the field of artificial photosynthesis. That is, if scientists can figure out how the slugs maintain stolen, isolated plastids to fix carbon without plant nucleus, then maybe we can harness the isolated plastids for energy green machines to create bioproducts or energy. The existing paradigm is that to make green energy, we need plants or algae to run the photosynthetic organelles. However, these slugs have given insight that hope is not the case to be. That this is not the case to be. Thank you for watching. This has been Biology in the News with Noah Smith.
Hello, and welcome to yet another segment of Biology in the News. I'm your host, Isabella Swatosh, and today I'll be discussing the use of Korean red ginseng and its effects in the healing process post-ischemic stroke. To begin, let's define an ischemic stroke. This type of stroke accounts for approximately 80% of those seen in stroke victims. In an ischemic stroke, there's a blockage of a cerebral artery, resulting in insufficient blood flow to the brain. When this occurs, the cells in this part of the brain affected begin to die. This is a stroke. When a patient suffers a stroke, the inflammation that results from the initial tissue damage is ultimately what causes the long-term damage strokes are notorious for. And while many have tried to find new therapies to combat tissue damage post-stroke, one team of scientists went back to their roots to find a remedy. Korean red ginseng, an herb widely popularized for its medicinal properties, was utilized in a study conducted by Lei Liu, among other authors. Mice were given Korean red ginseng via gavage every day in the 7 days leading up to their induced ischemic strokes. The team hypothesized that when mice were fed this herb, the subjects would show improved long-term recovery after the controlled injury. After strokes were induced in each of the test subjects, the recovery process was monitored over a 28-day time period, focusing on sensory motor deficits. And after a lengthy observation period, the results were in. The mice pre-treated with the ginseng showed not only reduced sensory motor deficits, but also long-term recovery compared to controlled mice. Well, improved long-term recovery. The initial stroke also had less of an effect on the pre-treated mice, as the lesion volume was found to be less so than the untreated mice. In conclusion, the team found that the ginseng can dampen long-term effects of ischemic strokes, as well as aid in the recovery process. Hopefully, in the future, this remedy can prove its usefulness in humans. Thank you for tuning into this segment of Biology in the News. Have a wonderful day and love life.
Hello and welcome to Ms. Johnson's 4-Odd Biology in the News. I am Megan Thomas, and today's topic is the frequency of low vitamin D3 levels in subjects with Parkinson's disease. Vitamin D, a fat-soluble vitamin, increases absorption of calcium, phosphate, and magnesium. Vitamin D3 are essential compounds in our body. Parkinson's disease is a neurological ailment of old age and unidentified causes. Vitamin D3 assumes a critical part in pathogens of skeletal ailments and calcium homeostasis. Vitamin D insufficiency predicts expanded danger of other perpetual ailments, malignancy, and cardiovascular illnesses. Constant deficient vitamin D values promote constant loss of dopaminergic neurons and propose to assume a major part in the pathogenesis of Parkinson's disease. Hundreds of populations who suffer from Parkinson's disease are vitamin D deficient. The connection among PD and D3 have a strong correlation in old age and fall risk of intimate additional search into the method essential for this connection. Subjects with Parkinson's disease have decreased vitamin D correlations in relation to controls. The recent research explores whether D3 levels predicts Parkinson's disease occurrence in populace of Pakistan, where solar exposure is high from different zones of the world. 243 diagnosed PD subjects from the local community enrolled during 2014 and 2016. Subjects with neurodegenerative thyroid ailments and new changes in lifestyle or nutritional status excluded. Educational station, routine daylight contact, cigarettes, alcohol, head injuries, pesticide exposure, and medical history were recorded. In the study, the average age of the participants were 67, with minimum of 56 years and a maximum of 85 years. Most participants were male, while others were female. 232 were married and 11 unmarried. 162 were from rural areas and 81 were from urban communities. By occupation, 27 had no occupation, 73 housewives, 100 manual workers, and 43 were office workers. 64 were uneducated, 123 primary to matriculation, 48 intermediate to graduation, and only 8 were postgraduate. A large number of study subjects, 213, had no evidence of Parkinson's disease in family, whereas positive family histories were 30 subjects. The risk factors for Parkinson's disease: history of pesticides contact in 9, smoking 61, alcohol abuse 24, and history of head injuries were observed in 18 subjects. Baseline characteristics of Parkinson's as tremors, bradykinesia, speech changes, impaired posture and balance, rigidity and writing changes were observed in 67%, 10%, 6%, 5%, and 4.9% respectively. Regarding duration of Parkinson's disease, 82 were less than a year, 41 1 to 2 years, 41 2 to 5 years, and 40 had a duration more than 10 years. 151 subjects of vitamin 25 OHD, less than 30 nanograms per milliliter, with 92 having normal vitamin D values, 30 to 50 nanograms per milliliter. Insufficiency, 10 to 30 nanograms per milliliter, were found in 148, and deficiency, less than 10 nanograms per milliliter, were seen in 3 subjects. The vitamin D3 levels were strongly correlated with age, duration of Parkinson's disease. These were statistically significant. The other risk factors were not a significant correlation of various risk factors of Parkinson's disease and vitamin D deficiency. Vitamin D deficiency is commonly associated in patients suffering from Parkinson's disease. and present research that as the age advances, risk of Parkinson's disease increases with simultaneous decrease in vitamin D levels. As concluded in the study, 62.1% of subjects of Parkinson's disease were vitamin D deficient. This has been Megan Toms from Ms. Johnson's 4-Odd Biology in the News. Thank you.
Hi, this is Eric Trung, and thank you for joining me in our last episode of Johnson's Biology in the News Network. Today's topic, we will be talking about xenotransplantation. Every day, the number of people who need kidney, heart, liver, and other transplants grow, and the supply of organs readily available are decreasing. Many people at the end stages of kidney disease die because the number of organ donors has drastically decreased. Scientists are now in the process of creating a practical way to meet these needs. Xenotransplantation may provide the solution to this problem. Xenotransplantation is the process of transplanting an organ from one species to another species. The process of xenotransplantation can theoretically provide an infinite source of organs, yet there are many barriers towards its practical use in the operating room. Now y'all may be wondering, what animal will we have to extract organs from? And they are pigs. Before implanting pig organs into humans, there are many major obstacles to overcome. One of the most important obstacles to overcome is the rejection of the organ inside the patient caused by human antibodies and the possibility of infections and viruses that are caused by the pathogens within the pig. Viruses are the most likely culprit for the rejection of an organ. Minimizing contact and by using immunization tactics may help or prevent the spread of disease from pigs to humans, but even at that, retroviruses may spread. Porcine endogenous retroviruses, or PRV for short, are RNA viruses that are encoded within the pig's genome and therefore may be passed down to their offspring. These types of viruses are capable of infecting human DNA as well. Several ideas have been proposed to lower the spread of PRV. Some ideas are raising a pathogen-free farm or vaccinating the patient before surgery. In a 2015 study, Yang and a group of researchers have used CRISPR technology to inactivate PRVs within the pig's genome. CRISPR, or clustered, regularly interspaced, short palindromic repeats, is used for gene editing, and the group used CRISPR to mutate a sequence of DNA at the loci, which affects the function and gene expression of PRV. Their results show that the ability for porcine kidney cells to spread PRV decreased 1,000-fold when coming into contact with human kidney cells. Now, in a recent study by the same group, they have planned to create pigs with inactive PRVs. They did this by designing guide RNAs within the CRISPR to mutate the sequence in the PRV loci. But this time, they included growth factors and apoptosis inhibitors so that the CRISPR can successfully mutate all the PRV. The CRISPR was then exposed to a cell line of a fetal pig, and a karyotype was performed showing that the sites where PRV sites were spliced out of by CRISPR. Pig embryos were then, with the least amount of chromosome deletions, were then transferred into a surrogate. On the 5th day of pregnancy, the fetus's DNA was sequenced and exhibited 100% inactivation of PRV sequences. The group then produced 37 PRV inactivated pigs. But then, sadly, they did not make it after 4 months. This study showcased a practical and possible realistic way for obtaining readily available organs that many people around the world need today. This study helped take down a huge barrier towards the practice of xenotransplantation. A lot of questions still arise, such as, is this even ethical, or is this 100% safe, or is this the right direction to go in? Well, that's all for now. Thank you for joining me in the last episode of Johnson's Biology in the News Network. There's always something new in biology, and it is our duty to share these new discoveries and advancements. Let all our curiosities grow. This has been your host, Eric Schwung, signing off.